Thanks.
Okay. My name is Merritt, and I'm the co-director for the Emory Stem Cell Core. We are part of the Emory Integrated Core facilities. As you know, our facilities are all over Emory's campus. So where we are. So we are actually located in Whitehead Biomedical Research Building on the 4th floor in 425. So briefly, we'll just go over the topics for today's talk. We'll review over what is induced pluripotent stem cell and how we reprogram these somatic cells to iPS. The current services offered by the core, new services we just started this year, and how you can use stem cells and iPS in your research. So first, let's review over what is an induced pluripotent stem cell and how we reprogram somatic cells to iPS. So what is a stem cell? For this talk today, I'll be referring, when I refer to stem cells, I'll be talking about human stem cells. They have 3 key properties. They're capable of dividing and renewing themselves for a long period of time. They're unspecialized, but yet give rise to specialized cells. There are 3 types of stem cells. We have embryonic stem cells. These are pluripotent stem cells that are able to differentiate into almost every cell type in the body, and they can renew indefinitely. We have induced pluripotent stem cells. These are derived from somatic cells. Again, they're pluripotent, and they can renew indefinitely under proper culture conditions. We also have adult stem cells. These are multipotent, meaning they can only give rise to related cell types. They do self-renew, but not indefinitely. So our core is mostly focused on the pluripotent stem cells. So we have 2 types, as I mentioned, embryonic stem cells, which are derived from the inner cell mass of a blastocyst. And then the other type is iPS. These are somatic cells that, using reprogramming factors of key reprogramming factor of the developmental pluripotency pathway to reprogram a somatic cell into a stem cell. Both of these cells are able to differentiate into a variety of cell types. Stem cell iPS cells are very similar to embryonic stem cells. There are subtle differences, including mitochondrial content, epigenetic status, and perhaps their X chromosome abnormalities. So there's a variety of source material that can be used for iPS. The 2 main types are dermal fibroblasts and peripheral blood. Here, these are fibroblasts that were derived in the core. These were derived from a skin biopsy. And then these are erythroid progenitors, which were derived from peripheral blood in the core. As you can see, they each have their own cell morphology. But using reprogramming, they both form an iPS cell. So here, I'm just going to briefly go over how we reprogram fibroblasts in the core. So we take cultured fibroblasts, we transduce them using a non-integrating sendivirus with the transcription factors OCT4, SOX2, CMYC, and KLF4, commonly known as the Yamanaka factors. The cells are then reprogrammed for up to 21 to 30 days. After reprogramming, total colonies are then isolated and then cultured for up to 10 days. This is to verify that the sendi transgenes are turned off and the cell is exhibiting its own endogenous pluripotency. We also isolate, expand, and reprogram EPCs from peripheral blood. Here, we take whole blood. This can be as little as 5 mils. Is needed to isolate the erythroid progenitor cells. Erythroid progenitor cells and CD34 cells actually have the highest efficiency for reprogramming, but they're actually in low quantities in whole blood. So we were able to take these EPCs and expand them for up to 17 days. That's to ensure that we have enough cells for reprogramming. The cells are then transfused using the same non-integrating sendivirus. And then, like the fibroblasts, they are reprogrammed. This reprogramming is a little bit quicker. It's about 18 to 30 days. Similar to the fibroblasts, we isolate clonal colonies and then they're cultured for 10 weeks. We also offer characterizing iPS. This is a two-part. We do one by immunofluorescence. The cells are fixed and stained for intracellular markers OCT4, SOX2, and nano. And cell surface markers SSEA4, Tri160, and Tri181. These images are then given back to the user for publication purposes or for future grants. The second part is RT-PCR. Total RNA is isolated from the clones. cDNA is then synthesized from that RNA. PCR reactions are then run for OCT4, SOX2, REX1, and NANO. And then a transgene-free status is confirmed with Sendai-specific primers and a Sendai-positive control reaction. OCT4, SOX2, and nano are all involved in the pluripotency lift pathway. And nano interacts with REX1. When cells tend to differentiate, REX1 will be severely and abruptly downregulated. So as you can see here, REX1 is very much present. We also offer karyotyping. This is actually a very important part. Karyotypical abnormalities can occur during reprogramming. And also during long-term culturing. It's important to have your cultures karyotyped after reprogramming, before experiments actually begin, and again after long-term culturing. Okay, so just quickly we're going to review over the current services. So as I previously mentioned, we do biopsy processing. We can do skin biopsies for fibroblasts. We will provide you with 10 vials of cells at least 2 different passages in addition to live cells. And then also the whole blood. With the whole blood, we actually isolate and will freeze back PBMCs. We will also isolate and expand the erythroid progenitor cells I mentioned, and then freeze those and hand those back as well. Obviously, we offer reprogramming to iPS. We guarantee a minimum of 6 clones, and then freezing of several vials, which will be handed out back to the user at the end. We also offer characterization, as I mentioned, which includes that IF confirmation and RT-PCR confirmation. As I mentioned, we also offer karyotyping, which will include a detailed report, which will be emailed to the user of any kind of abnormalities or normal karyotype, so that can also be used for publication or grant purposes. Additional services we offer is the thawing of fibroblasts. We will hand back live cells as well as up to 10 vials of frozen cells. This would be at least 2 passages. We also offer thawing of iPS for embryonic stem cells. We will hand back the live cells. This will also include at least 3 passages of growth and at least 2 passages of frozen cells. We also offer training and consulting. So you can learn the basics of iPS in your own lab, so I can actually come to your own culture room. Additionally, I can offer evaluation of your cultures, protocols, techniques, and this is done on an hourly basis. So today I'd really like to talk about some new services that are being offered by the Core. Lymphoblastoid cell line generation. LCLs are commonly used as an unlimited resource for various genetics and immunological studies. So from here we take whole blood, we isolate the PBMCs, and then we add media containing Epstein-Barr virus for immortalization. The cells are then grown for 2 to 3 weeks. We then hand back cultured cells and frozen vials. And our second new offering is neural progenitor cell generation. Here we take iPS cells, and they're plated and then put into a defined medium containing SMAD inhibition. The cells are then passed for up to 21 days. Then they're considered P0 EPCs. The medium is then changed to an MPC medium. The cells are then grown for 3 more passages. At day 42, they would be handed back as live cells. and are ready for further differentiation into the neuron subtype of your choice. After P0, we actually will stain and confirm that they are EPCs. At P1, we will freeze back 10 vials. Those will be returned to the users. And then after PT, we'll isolate RNA for further use. And then here's just some of our plating density, the media, and the substrate they're grown on. So here I want to talk about how IPS that were generated here at Emory are currently being used to make EPCs and for further neuronal differentiation. This is courtesy of Dr. Raymond. He is a postdoc working on 3Q29 schizophrenia projects. So here are some IPS that were generated in my lab. The blood was actually isolated from patients at the Marcus Autism Center. I then took it, isolated the EPCs, and then reprogrammed the cells to IPS. They were then induced into neural progenitor cells. So you can see that the cells were then stained and show some very defined MPC, including neural rosettes. And then they were further differentiated into foregoing neurons, as indicated by the last pictures, showing some common foregoing neuron markers. So how do you start a project with a core? Email me. So you can email me and we can discuss initiating a project. We actually use PPMS, which is great because it provides you access to our services. And you can also track your projects in PPMS. We have a website, which is always available. You can find all the information about the services we offer, as well as the pricing with the services. Okay, so last but not least, the use of stem cells in your research. So IPS differentiation, as I mentioned previously, there is I'm sorry, for some reason it's not coming out. Oh, there it is. Okay, so here's just a general snapshot of the just kidding. It was a general snapshot of the ability for IPS to turn into a variety of different cell types using using defined medias and different protocols. So stem cells can also be used in basic and translational research. As we mentioned, we take somatic cells, induce them into pluripotent stem cells, and then form specialized cells. These specialized cells can then be used for disease modeling. They can be used for drug discovery, toxicity studies, and then eventually cell therapies. Another big use of stem cells in the last couple of years has been organoids. These are 3 cultures of stem cells that actually recapitulate a simplified version of organs. Now you can form intestinal organoids, hepatic organoids, neural organoids, and pulmonary organoids. And these organoids can then be used in research as well, such as disease modeling, drug testing, host microbe interactions, proteomic studies. And then last but not least, IPS is a common source for material for gene editing, which Dr. Raymond will actually discuss after this. So quickly, just my acknowledgements are the Emory School of Medicine cores, the Department of Cell Biology that actually hosts us, the Laboratory of Translational Cell Biology, which is where I came from before and developed several of these protocols, and the 3Q29 project, in particular, Dr. Ryan Purcell, for letting me show you some of this work today. So I will let Chris give the next part.
All right, thank you, Megan. My name is Christopher Raymond. I am Associate Director for Core Innovation for the Emory Integrated Core Facilities. So today I'm going to talk, as Megan mentioned, about cell line engineering. So once you have your iPSCs, or maybe you have just a regular cell line, perhaps you want to make a disease model out of these. You may want to make a mutation or tag a gene in it. And we recognize that it may not be particularly efficient for you to do this in your own lab. So we are now looking at providing the Emory Integrated Core facilities are now looking at providing cell line engineering as a service. So just briefly, as I'm sure you're aware, CRISPR-Cas9 technology has been extensively in the news, and even recently with the infamous work of He-Jun Koo and the human germline gene editing. So certainly this is a nuclease-mediated gene editing has been actually around for the last 12 or so years, at least in animals and in cell lines. First with the use of zinc finger nucleases, and then with talons, and now with CRISPR-Cas9. All those techniques actually work well for gene editing, just that CRISPR-Cas9 is so much cheaper and easier to manufacture. It really makes gene editing accessible to the masses, per se. Just a brief overview of how this technology works. Of course, CRISPR-Cas9 it's a ribonucleic complex. You have a Cas9 protein here, which is bound to what's called a guide RNA. This guide RNA has a specific has a specific target sequence that will recognize at your DNA. This is also adjacent to what's called a PAM site, which is required for it to target to that loci. So this RNP causes a double-stranded DNA break at the target location, and then the repair of this double-stranded break in the cell is either repaired by one of two pathways, and that is the NHEJ, or the non-homologous end-joining repair mechanism, or the homology-dependent repair. So you can either, in the presence of a donor sequence, it can then actually use this donor sequence to replace a sequence at that DNA break site. Now, CRISPR-Cas9 is not a panacea. This mechanism is actually much more effective at, CRISPR-Cas9 is much more effective at breaking things than it is at making things. So making just a small mutation, an indel or insertion deletion mutation, is much more efficient than inserting a small tag. In fact, the larger sequence of insertion, the actually less efficient it becomes. So small point mutations are fairly easy to do, and even small tags, but then large insertions are much more difficult. Overall, the cell would use the NHEJ pathway maybe greater than 95% of the time. So in fact, you don't get this type of the cell does not utilize the homologous-dependent repair very efficiently. We've been making actually mouse models here at Emory using CRISPR-Cas9 technology since about 2016, and this is done by injecting your guide RNA and your Cas9 directly into a one-cell embryo, and then with or without a donor sequence, depending on the mutations we made. And we've generated about 50 different mutant mouse lines using this technology. So now we want to take this technology from the mouse and bring it to the cells, either regular cell lines or iPSCs. What we see now as happening is essentially a collaboration between Megan Stem Cell Core and the Genomics Core to provide Emory investigators interested in genome editing of cells of any cell, any type. We eventually will be located in the new HSRB2 building. However, that won't be for still several more years. And so we also now have some primary funding to get this technology up and running from the Woodruff Foundation. All right, so we're going to take two different approaches for using this technology at Emory. And one is, first is to develop our internal capabilities. The second one would be to leverage existing, say, outsourcing facilities. And I'll show you how we'll integrate the two. So right now, the core of the collaboration looks something like this. So I was actually formerly with the transgenic core, and my background is in genetic engineering, so I do a lot of the design of the guide RNAs and donor sequences. Megan is just heard from at the Emory Stem Cell Core, is going to be working on all the transfections of whether it be an iPSC cell line or just a regular cell line. And all the genomics work will be done over with Lyra Griffiths at the Genomics Core. So right now we're currently doing a knock-in pilot experiment, which is ongoing. And I'm just going to show you now. This is just kind of a generic workflow of what it looks like to do CRISPR cell line engineering. It involves the steps of host cell characterization, just making sure karyotypes looks good. They're not mycoplasma positive or something like that. Then of course the guide RNA design and maybe donor design. Then you have a point where you need to do the actual transfection or generate a pool of cells that have been transfected with the Cas9 and the guide RNA, and with or without a donor. And then these cells need to be analyzed and then plated for single cell cloning, and then again genotype till you get a clone with your desired mutation. All in all, this has already been around 4 months for workflow. I think it's something reasonable. Probably more than 4 to 6 months would be probably the real time for generating a knockout cell line.
What would you see a success rate is it 100%?
For us, too early. We're just starting at the initial work right now. So there's a number of ways you can deliver Cas9 into the cells, either by plasmid or virus, or just using a synthetic guide RNA Cas9 protein itself. Really, the state of the art right now is to use the RFPs, and there's a number of reasons. I actually don't say it's not actually a high cost. It's actually these costs come way down now, and I call it a low cost. The advantage of this is that it's really good for both, again, just regular cell lines, iPS cell lines, primary cell lines. Good for, you can use it with lipofectomy transfections or electroporations. This complex is not integrated, very low toxicity. It's very high turnover, so you get very low off-target effects. So really, it is the best way to go now for gene editing. In order to help us assess gene editing, we have recently acquired a QX200 Droplet digital PCR system from Bio-Rad, which is now housed in the Genomics Core over in the Woodruff building. What this does, it's a very nice technology. It will enable us to take a cell pool that's been transfected with guide RNAs and Cas9 and be able to assess the percentage of cells that have a mutation, either just an indel or in case of, say you're making a SNP, the percentage of knock-in in that cell pool. And that will now enable us to really determine how many plates that we need to make in order to get a single cell clone out of that transfected cell pool. So this really cool technology, it's basically like regular PCR, but instead of just having one PCR reaction or tube, it actually takes your PCR sample and actually disperses it into droplets, about 20,000 droplets per tube. And you're able to see that in some of these, depending on the quantity of your target, some of these droplets will actually amplify in some of them and not in others. And so it actually quantitates your PCR product, and you're able to determine the number of the percentage of cells that are of target cells that you want. This droplet digital PCR is actually very good for it. It has just a number, just besides the CRISPR work that we're doing, it has a number of applications. Whenever you want to do a quantitative PCR, it's much more, I guess you'd say, truer readout than just an TaqMan type assay. As I said, this technology is now over, or this machine is now over in the Genomics Core, and I think will be available for investigators for use for other applications. Just an example of, here again, this is just a work that's done by Megan and Lyra. This is, again, one of our pilot studies. You can just see here, you can design different assays here just to measure the amount of cells that are undergone an indel mutation, or NHEJ here is labeled. You can see that wild type cells, as shown in the upper right-hand box, is a cell population which is labeled by two different probes, and the bottom box here, which identifies a population which is only recognizes by a single probe, as the other probe has been lost due to mutations at a specific site. So it's able to quantitate out of this population of cells what that population of gene editing in that cell pool is. Likewise, you can take a also looking at we can quantitate the amount of knock-in, basically if we're making a knock-in mutation in this case, looking for a point mutation. And again, you can label probes and identify cell types, where you can look at here. This is a wild type population, a population that has indel mutation, then a population which has knock-in of your gene of interest. So it's very good technology, and it's going to be very helpful for us to generate knock-in or knockout cell line clones. Okay, so now I'm going to talk about a little bit about leveraging external expertise, and specifically I'm going to talk about partnering with a company called Synthego. For those who are not familiar with Synthego, they've been around for about 4 or 5 years, and I've been working with them with the mouse CRISPR applications. They are a company basically a company of engineers, SpaceX engineers, that formed a really an automated gene editing company with automated gene editing. A company with gene editing company with automated capabilities, and so basically all their processes are automated and really do not touch human hands. So it allows them to generate cell pools or cell clones very efficiently. And so they can actually provide knockout or knock-in cell pools with a guaranteed 50% knockout or knock-in efficiency, and oftentimes this can be 70, 90%. So the idea here would be that we could actually leverage their ability to make knockout cell pools, and then provide a single cell cloning service here with the Emory core. Just to give an idea of their capability, they actually take all their cell lines and optimize their transfection conditions. So various cell lines transfect differently. You can see in the top left-hand corner these EMT6 cells are very easy to transfect, and these other two at the right are less amenable to transfections. In fact, the one on the right here, these Kasumi3 cells, only show really very few conditions that actually work to transfect these cells efficiently. So they're able to simultaneously electroporate with 200 different conditions and then analyze the knockout to really get these efficiencies up. And currently they have over 150 human cell lines that are available through the company, as well as a number of mouse lines and iPS cell lines as well. And customers can actually supply their own cell line as well for a little bit extra cost. So what do you get from them? So knockout cell pools, they can deliver a knockout cell pool, which is just 2 vials of edited cells. They can ship those in as little as 3 weeks, so because they're very fast and automated, and at cost of actually this is now down to $2,500. So very, very cheap and effective. If you want to make that a clone, though, that price goes up to actually now $12,500. So about $10,000 more to make a clone with Synthego. And so we find that difference there, that 10,000 difference, that we would be able to take that cell pool and actually bring them to single cell clones for a cheaper price than they do at Synthego. And again, they can do knock-in cells as well. Either SNPs, codon changes, small tags, small insertions, and pools there do go up because, again, like I said in the beginning, it's much less efficient to generate knock-ins than it is to generate knockouts. And again, around 10,000 or so for small insertions, and about 25,000 for larger insertions. And this is actually clones for that. So again, about 10,000 or so more for making the clones. And as well as they have developed a very efficient iPSC knockout and knock-in platform as well. And again, you're getting two vials of clones each with this service. Again, knockouts and knock-ins taking about pools at around less than two months for that. So very quick for that. So this is I'm going to summarize here the pricing for each of these. So you can see that in just regular cell lines, a pool now runs about $2,500 for a knockout, and then 10 to 15,000 for a pool of knock-in cells. And again, cloning can be anywhere from 10 to 20,000 more for these, and that's why I think we have the opportunity to provide the service at a much cheaper, or the cloning service at a much cheaper cost. Again, and also because our iPSCs our platform is actually pretty efficient, they're able to provide pretty good pricing compared to other companies in terms of their iPSC gene editing as well. So I think that concludes my section. If you have any questions, happy to take them, or Megan as well. Thank you.
Just give the people online a minute to see if they have any questions. I'm not seeing any, so any questions from the room?
That was pretty clear. Thank you.
Thank you.
Yeah, thanks very much for
Thank
coming
you.
to do this.
Thank you.
Thank you
Oh,
guys.
wait a minute, 2 questions came through. Is there a difference with iPCs derived from fibroblasts versus PBMCs?
you want me to
Yeah, if you go up there.
Yeah, so they can hear me. Just repeat.
And if you would repeat the question.
Yeah,
too.
sure. Yeah, so the question was whether there are differences between iPS derived from EPCs and fibroblasts. So there tend to be, research is now showing that the original source does kind of impact the differentiation potential and some of the epigenetic status. So if you are looking to differentiate into a specific cell type, it's easier if you start with whichever cell is in the same lineage. But I'm more than happy to go over that with anyone if they have particular questions.
The other question is simply, will it be possible to receive a copy of the recording?
I assume yes. If you signed up for this webinar, I believe you'll receive a copy of this recording.
Or do you want them to email you if they want a copy, or?
Is it anyone that registered within the week?
Okay,
They should receive it within a week.
that's it from online.
Okay.
All right,
Thank
thank
you.
you. Thank you.
Yeah, I think I hit stop.
Oh, I had the gene editing question actually. So what time, at what point in the difference